Good morning. We're going to pick up with the second video on chapter 10. This video specifically is going to talk about mitosis and cytokinesis. Mitosis is the division of the nucleus in a cell that is dividing, and the cytoplasmic division or splitting is called cytokinesis. Okay. So, we're going to pick up the cell cycle. And we're going to pick up after interphase. The first step in the division of the nucleus that contains the condensed chromosomes is called prophase. There's a list of events that happen within the cell. The chromatin condenses to form chromosomes. The protein filaments or tubules called microtubules begin organizing and forming a spindle, a spider web-like structure for these chromosomes to split apart on and migrate on. The nuclear membrane dissolves where these chromosomes are contained within. The spindle attaches to the chromosomes. This region within the centromere actually is called a kinetochore. So the figure you see on the left, kind of light blue, you see a dividing onion cell that's kind of square or rectangular, and you see the reddish purple chromosomes. The purple outer circle or oval is part of the nuclear membrane, and you see it's kind of breaking apart. On the right, you see a photograph of a fish cell, and you notice how circular the cell is, and the blackish strands in the middle of that cell are the condensed the chromosomes. So that kind of gives you an idea of what a plant and an animal cell going through cell reproduction in the stage called prophase, what that looks like. So prophase is the first step in the division of the nucleus. Prometaphase is considered to be a late prophase, early metaphase portion of the cell cycle. And the main thing that happens during this step is the chromosomes attach to the spindle fibers. Again, these spindle fibers are part of the cytoskeleton of a cell, and they're made of the protein tubulin. They form a spider web-like arrangement, and they provide a pathway for the chromosomes to travel and end up in separate daughter cells at the end of the process. So prometaphase, the chromosomes attach to the spindle fibers in a region of the centromere of the chromosome that's called a kinetochore. And that's what is illustrated, or attempted to illustrate anyway, with these two photographs. I thought I would put up a picture of the spindle. Some people call it a spindle apparatus. Some call it a bipolar spindle because it has two poles, if you will. The poles are illustrated and labeled in this particular illustration. The spindle fibers are made up of microtubules. They're a part of the cytoskeleton, again, that has multiple functions, including cell reproduction. This particular illustration shows the chromosomes, of which there are 4, 2 purple and 2 blue. They are lined up in the middle of the cell, and you notice there's a little white region on each chromatid that's called the kinetochore, where the red spindle fiber is attached to. So the bipolar spindle enables the duplicated chromosomes, the chromatids, to separate and migrate to opposite poles of the dividing cell. So it's a very important structure in cell reproduction. Metaphase is when the chromosomes have migrated along these spindle fibers, and they have migrated to the, to the equator, the, if you will, equatorial plane, equatorial center of the cell. This is where the cell will actually split in half. The cytoplasm will split in the area where the chromosomes align during metaphase. You see both metaphase in the onion and metaphase in the fish. You can clearly see in the fish cell the region of the centrosomes or the centrioles, the opposite poles where the microtubule fibers originate from. You can really see that on either side of the blackish region of chromosomes in the middle of that cell. So where the chromosomes align, the equator, if you will, that is where the cell will split in half, or the cytoplasm will divide. Okay, so P for prophase and prometaphase, M for metaphase, A is for anaphase. In anaphase, the centromere region replicates. Each sister chromatid then begins to pull apart. We refer to them then as chromosomes. Each chromosome then is pulled to the opposite pole. The spindle fibers contract and they pull the chromosomes toward the opposite poles of this dividing cell. It is usually considered that during anaphase, the cytoplasm will begin to divide. The cytoplasm begins to divide in the equatorial plane, the equatorial center of this cell. Because each cell replicated its chromosomes in interphase, each of the eventual daughter cells will contain the same number of chromosomes if a mutation has not occurred. The example shows a dividing cell with 4 chromosomes, and during anaphase, each of the eventual daughter cells will have 4 chromosomes. So mitosis is a cell reproduction event in which each of the daughter cells has the same number of chromosomes as the initial parent cell. So again, you see the onion on the left and the fish cell on the right. You see the chromosomes being pulled apart and moving toward opposite poles of the cell. The last portion of mitosis is telophase. Each cluster of chromosomes reaches the pole. A nuclear membrane will develop around these clusters. The chromosomes will decondense and form chromatin, long stringy strands of DNA that basically unwraps around the histone proteins. The spindle fibers will disappear. They're no longer necessary. And you have a not a daughter cell, but you have a parent cell that has 2 nuclei within it. So now we need to talk about how does the cytoplasm end up separating to form 2 daughter cells. This is called cytokinesis. So at the end of telophase, you have 2 nuclei formed within a parent cell. So the nucleus has divided. The initial one in the parent has now divided to form 2. So cytoplasmic division now, how one parent cell will split its cytoplasm to form 2 identical daughter cells. You see the process of the cell cycle in the fish. The top left is interphase, top right is prophase, metaphase, middle, is the bottom left. In the middle, on the bottom, is anaphase, and you see telophase in the bottom right. And you notice that the parent cell is beginning to pinch in half, and the cytoplasm is splitting, if you will, cytokinesis. A little bit harder to see each particular stage in a plant cell, but this is showing you an onion. The majority of all the cells in the upper left photo of interphase, they are in interphase. Prophase is labeled by that one cell that is lacking the nuclear membrane. You can see down below prophase in that upper right illustrate or photo that there's a cell in metaphase. Metaphase is also shown in the lower left photo. The lower middle photo shows you a cell in anaphase, and right above it is a cell that has just finished telophase. And cytokinesis, you can see a very faint cell wall in between those two nuclei. You also see a prophase in that particular photo. In the last illustration, the lower right, you see telophase. You see several plant cells there, onion cells that have gone through telophase. So, in telophase, when it finishes, it's usually considered that the cell has split its cytoplasm to form two daughter cells. We refer to cytoplasmic division as cytokinesis. It's usually not given its own name. It's usually considered to be part of mitosis, beginning in late anaphase and completing by the end of telophase. It is a little bit different in plant and animal cells, primarily because plant cells have a cell wall. So I'll talk about plant cells first. In the plant cell, cellulose begins to be deposited about at the midpoint of the parent cell, and then the cellulose is deposited toward each of the walls of the parent cell. And basically the cellulose will form a cell wall between the two eventual daughter cells. This is referred to as a cell plate. So the cell plate helps to direct the deposition of cellulose to form new cell wall material and form two daughter cells. And animal cell and also our cells divide by what is called a contractile ring or a cleavage furrow. The single oval cell is basically split in half or pulled in half by a portion of the cytoskeleton that is referred to as actin filaments or microfilaments, if you will. So a band of actin filaments begins to pull the parent cell in half to form two daughter cells. The last slide kind of gives you some illustrations of cytokinesis in an animal cell and a plant cell. So the animal cell shows that the equatorial region where the cell began to split from where the chromosomes were lined up at metaphase, that the animal cell has a ring of actin filaments, microfilaments, that will pinch the parent cell in half to form two daughter cells. The plant cell, which is more square or rectangular in shape due to the cell wall, a cell plate forms typically in the equatorial region again where the chromosomes were aligned in metaphase. A cellulose, the main material in the cell wall, is laid down from the middle of the cell working out toward each end. And when that is completed, you basically have two new plant cells, two daughter cells. Since we're referring to mitosis in this process, each daughter cell is typically genetically identical to the initial parent. So the chromosome number does not change. It remains the same in each daughter cell. This occurs hundreds of millions of times every minute in the human body. So mitosis is a very common process of cell reproduction in the human body. In chapter 11, we will pick up with meiosis, the formation of gametes. Thank you. Bye.